Today we will discuss natural biological assemblies at nanoscale, which is our main topic. So what is basically the biological assembly? Biological assembly is something that is referred to as the biological unit at the micromolecular assembly that has either been shown or believed to be the functional form of the molecule. So what are the characterization of natural biological assemblies at nanoscale? That assembly should be well-organized architecture with a broad selection of sizes at the nanometer scale, monodispersed particles with uniform size and shape. There could be variability of genomic sequences through which the composition and surface properties could be controlled through recombinant technology. And the last one is economical. Large-scale production in grams or even in kilogram quantities could be performed. So first of all, at nanoscale we will discuss self-assembly of DNA. First of all, manipulation of DNA. First response is strand displacement. This displacement is very well organized of the single strand of DNA from a double helix by an incoming strand with a longer complementary region to the template strand, and there is always a template strand. The second well-organized function is DNA restriction, that is basically the cleaving of phosphodiester bonds between the nucleotide subunit, and that always occur at a specific location by a short 4 to 8 base sequences by a class of enzymes which are known as nucleases. If you have some restriction nucleases, they will cut only at the specific site. And same, if there is nicking then that always occur at the same position. Third assembly process is DNA ligation, that is basically the rejoining of nicked double-strand DNAs by repairing of phosphodiester bonds between the nucleotides by a class of enzyme which is known as ligases. Next process is DNA polymerases that catalyze the polymerization of nucleotide triphosphate into a DNA strand. Deoxyribosomes are class of enzymes or nucleic acid molecules that possess enzymatic activity. For example, they cleave the specific target nucleic acids. Why use DNA to assemble molecular scale devices? Why you use DNA to assemble molecular scale devices? Because a variety of geometrics can be achieved by carefully programming DNA sequences to interact among themselves in a predictable manner. Second advantage is the structure of most complex DNA nanostructures can be reduced to determine the structure of short segments of double-stranded DNA. Third is you can design DNA nanostructures can be assisted by any kind of softwares. Fourth advantage is that solid phase chemical synthesis of custom single-stranded DNA is now routine and inexpensive, and assembly of DNA nanostructures is a simple experimental process. So next advantage is assembled DNA nanostructures can be characterized by a variety of techniques. Self-assembled DNA tiles and lattices. For example, DNA nanostructures, they could be stem loop, a single-stranded DNA that loops back to hybridize on itself. Second is sticky ends, an unhybridized single-stranded DNA that protrudes from end of a double helix, as you can see in this B picture. Here is the stem loop. Here in figure A, this has formed the loop-like structure. Holy day junction. Here is two parallel DNA helices form a junction with one strand of each strand. Here you are using the junction from one strand and another from the other DNA helix. Our DNA tile is a DNA nanostructure that has number of sticky ends. Here you can see that on one side, which are termed as the pads. Our DNA tile lattice is a DNA nanostructure that is composed of DNA tiles that assemble together via hybridization of their pairs. Here you can see these kinds of pairs. DNA origami, that was the first proposed and implemented by Paul W.K. Rudiment in 2006, in which he folded a long viral single-stranded DNA molecule to create DNA structure of any arbitrary shape. The term origami refers to Japanese folk art of folding paper in a special shape. Method is based on folding of large single-stranded, usually 7.3 kilobases genomes with an excess of smaller complementary strands. These small strands are called staple strands and are complementary to at least two distinct segments of long single-stranded DNA. Here you can see schematic illustrations of DNA folding and origami constructions. This is the two-dimensional DNA origami. This basically cube-like structure hollow box is indicating three-dimensional DNA origami.